Hello, everyone, and welcome to today's live webinar, 3D Organoid Models for Differentiation of PSC-Derived Dopaminergic Neurons. I'm Christy Jewell of LabRoots, and I will be your moderator for today's event. Today's educational web seminar is brought to you by LabRoots and sponsored by Thermo Fisher Scientific. For more information on our sponsor, please visit the Sponsor tab on the left side of your screen. Now let's get started. I would like to remind everyone that today's event is interactive, and we encourage you to participate by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click Send. We will answer as many questions as we have time for at the end of the presentation. If you have any trouble seeing or hearing the presentation, click on the Support tab found at the top right of your presentation window, or use that Ask a Question box to let us know you're experiencing a problem. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located at the top right of the presentation window and follow the process to obtain your credits. I would now like to introduce to you today's speaker, Dr. Richard Josephson, Senior Staff Scientist, Cell Biology 3D Models at Thermo Fisher Scientific. For a complete biography on our speaker, please visit the Biography tab at the top of your screen. Dr. Josephson, welcome. You may now begin your presentation.
Thank you, Christy, for that introduction. My name is Richard Josephson. I'm here to talk to you about 3D organoid models for differentiation of pluripotent stem cell-derived dopaminergic neurons. So, as you're probably aware if you're watching this seminar, in the past few years, there's been a boom of publications on organoids and other 3D culture systems for studying the human brain. Here at Thermo Fisher, some of our customers are trying to catch up with that boom, and so they're asking us some introductory questions, such as, how do I get started culturing organoids? And are 3D models really better than 2D? And how do I model my particular research area in organoids? So the work I'm going to discuss today was undertaken in response to those sort of customer inquiries, but it focuses on a much narrower question, namely, can Thermo Fisher's existing 2D culture systems be used to generate midbrain organoids and to model Parkinson's disease? As you're probably aware, Parkinson's disease is a progressive neurodegenerative disease in which a specific type of neuron, the A9 dopaminergic neuron of the substantia nigra, is lost in large numbers, while other neurons are largely spared. Nature sells a set of 3 media which have been optimized to specify, expand, and mature human dopaminergic neurons from pluripotent stem cells in 2D culture. This is the PSC Dopaminergic Neuron Differentiation Kit shown at the top of this slide. At the top of the chart is the workflow that's been optimized for 2D differentiation. Specification occurs in attached culture on vitronectin, followed by 2 passages onto laminin. At day 16, the dissociated cells can be cryopreserved if you wish. After day 16, the floor plate cells are expanded in suspension culture for 5 days and then passaged onto poly-D-lysine and laminin for the final maturation. At the bottom of the figure is an alternative workflow that my colleagues and I at Thermo Fisher have worked out to form midbrain organoids in 3D. I'm going to walk you through this procedure in detail, but for now I just want to point out a few highlights. First, we're beginning by culturing the pluripotent stem cells themselves in 3D in a new suspension medium. The cells are passaged only once instead of several times, right at the beginning of differentiation to reduce any irregularity in the size of the spheroids. And of course, the cells are kept in rotation or in suspension in static culture for the duration of differentiation. As I'll show you, we have found that specification and expansion of floor plate progenitors can be completed in a much shorter time in 3D, 11 days in the bottom workflow versus 21 days in the 2D workflow at top. Both methods produce a similar purity of floor plate progenitors, while also promoting the functional state of the resulting dopaminergic neurons. So here's the data on the floor plate specification efficiency. We are using two markers, antibodies to the nuclear factors FOXA2 and OTX2, to mark progenitors that have a midbrain floor plate identity. Cells that you see here have been passaged and replated as monolayers, either after 21 days of differentiation in 2D on the left column, or 11 days of differentiation in 3D in the right column. Antibody staining reveals that in either case, greater than 90% of the cells co-express FOXA2 and OTX2. Thus, 3D specification reaches the same efficiency as 2D specification, but in much less time. This next figure shows what happens if you take those cultures that have been specified and expanded in 3D and then replated in 2D out a little further. So with the standard 2D differentiation protocol, you go out to day 35, 5 weeks of culture, and then you can stain for Th-positive neurons. Whereas with the 3D protocol, when you dissociate at day 11 and plate them, by day 25, you see abundant Th-positive neurons. And if I zoom in a little bit, you can see that the Th-positive neurons in both cases are positive for the midbrain marker FOXA2. So to go back to the workflow slide just for a moment, I want to zoom in a bit on the details of this and talk about a number of variations that we've tried to the protocol in order to optimize differentiation of dopaminergic neurons in 3D. So first, zooming in, the input, as I mentioned, is spheroids of iPSC cells grown in rotating culture. These are generally passaged every 4 days, but we have found that the clumps 1 day after passaging are generally the optimal size for switching to the floor plate specification media. So that is what they look like on day 0 when we switch to specification. After 2 days of specification, you can see that there are outgrowth of these thin epithelial sheets of cells on the sides, which by day 6 are beginning to greatly thicken. You switch these cultures into expansion media at this point, and that allows the spheroids to grow up into these irregular shapes with a translucent outer layer, and those keep growing in size until week 3 or 4 of differentiation. As an alternative to this, we embedded some of the spheroids in extracellular matrix early in differentiation. This is a very commonly used protocol for neural organoids. We embedded in cold gel tracks at day 2 or at day 7 and see an immediate response in the formation of these little bubbles of epithelium on all sides of the organoid. By 2 weeks of differentiation, the embedded organoids seem to consist of convoluted sheets of cells. And by 4 weeks, that complex structure remains, but cells and neurites can both be seen growing out into the capsule of extracellular matrix. We also explored another set of alternatives in hopes of producing more uniform organoids. This time we passaged the iPSC cells into low attachment round bottom 96-well plates which are called non-clone Sphera plates. We plate cells evenly to all wells, and 1 day later, we get a single small spheroid per well of very consistent diameter across the plate. That's day 0 for the floor plate specification. These small spheroids respond to the addition of specification media by growing out bubbles of epithelium, similarly to what we saw with the ECM embedding. But this time, switching into expansion media allows those epithelial bubbles to grow outward without any constraint. And by 3 weeks, these have thickened to give very irregular shapes, but at least the sizes of the organoids are somewhat more uniform. So we decided once again that we should add extracellular matrix during the specification. However, this time, instead of embedding the early spheroids at day 2, which is very labor intensive, we simply added 2% Geltrex to the medium and kept it cold so as not to allow it to gel. At the end of the specification period, these organoids don't look bubbly at all. But when you put them into expansion media, you can see that they do have many thin, translucent bumps of epithelial cells. I'll show you in a bit that the convoluted outer structure is retained through further differentiation. So it was a little hard for us to believe that dilute extracellular matrix could really change the epithelial structure of these floor plate spheroids. Here we looked at spheroids after 1 week of differentiation in the U-bottom wells with or without dilute extracellular matrix. And looking just at the DAPI-stained nuclei, we see that 200 micrograms per mil of mouse laminin induces a major outgrowth of these hollow bubbles of cells, while 2% Geltrex does pretty much the same thing for less money, although it's a little bit less striking. We also looked at the structures by staining for the epithelial polarity marker NCADH. And we saw that extracellular matrix partially altered the distribution of NCADH into the lumens of these little bubbles or rosette-like structures. But it's not a completely black and white change. Next, to see how these alternative workflows affect neuronal differentiation, we compared the organoids at 3 weeks by Wholemount immunostaining. The organoids were stained with antibodies and cleared with the cytovista reagent, and then imaged on the Cell Insight CX7 laser high content analysis instrument. So the first example here is from free-floating organoids specified in rotating culture. At 3 weeks, these have abundant MAP2 positive neurons around the outside in red, while the tyrosine hydroxylase positive dopaminergic neurons marked in green are a subpopulation with relatively short projection. By comparison, when you embed these same organoids in extracellular matrix, you get a larger organoid at 3 weeks that has a large subset of TH-positive dopaminergic neurons. Those DA neurons also have longer projections than we saw in the free-floating organoid, and so could be more mature. You can also see that those convoluted cell layers are maintained, particularly when you look at a single optical section. Now, the organoids that were specified in static culture in a U-bottom plate look at 3 weeks a bit like the embedded organoids. DAPI shows that the structure is less compact than the free-floating organoids and features a population of elongated TH-positive neurons like the embedded organoids. Finally, the addition of dilute Geltrex to the static organoids produces a more regular shape that gives a convoluted outer layer with abundant Th-positive neurons. I don't have MAP2 staining available on this yet, but I will soon. When looking at a single optical plane of this organoid, you can see how the dopaminergic neurons have cell bodies that are distributed across that deeply crevassed surface. This is also visible in the next slide, which is a 3-week-old organoid formed in a U-bottom well with Geltrex and stained for TH and the midbrain progenitor marker FOXA2. You can see how the dopaminergic projections cover the surface, but Th-positive cell bodies can also be seen at the surface or deep below it. This is more obvious when you look at the single optical section on the right. This makes it difficult to see whether the progenitors and the postmitotic neurons occupy separate layers in these very wrinkly epithelia covering the surface. So now I want to talk about maturation of the dopaminergic neurons. Dopaminergic neurons in the brain produce a pigment called neuromelanin. It's a direct byproduct of dopamine synthesis. And neuromelanin is the substance that makes the substantia nigra black. Several recent publications on midbrain organoids have noted that neuromelanin appears in their organoids after 100 or more days of culture. So it's perhaps not surprising that our ECM-encapsulated organoids have dots of pigmentation after 9 or 10 weeks of differentiation. However, the unencapsulated or free-floating organoids show very little pigment in the same time frame. You may also have noticed that the encapsulated organoids by 10 weeks have become much larger. This appears to be because that capsule of Geltrex is completely filled up with cells and cell projections. Now, dilute 2% Geltrex added to the U-bottom plate also promotes neuromelanin production in the organoids. We can see the pigment easily at 9 weeks, but oddly enough, we also can detect that pigment in most of the organoids by only 5 weeks. So far, the dilute Geltrex in the U-bottom well is the only treatment that produces neuromelanin as early as 5 weeks. So we see additional evidence of early maturation in these organoids when we take them to record electrical activity. So these are pictures of midbrain organoids that have been plated onto multi-electrode arrays from Axion Biosystems. In the Axion plate, each well contains 16 electrodes for recording electrical activity. You can attach whole organoids to these, although as you can see in the pictures, it's difficult to exactly cover all 16 electrodes. These organoids were plated at 3 weeks before the neuromelanin was apparent. Then it took about 2 weeks before we could record the first bursts of spontaneous electrical activity. The bursts were sometimes recorded by only a single electrode and sometimes coordinated across 4 or 5 electrodes. And in these raster plots, the coordinated bursts are indicated by a vertical pink line. Because dopaminergic neurons can reuptake their neurotransmitter via the dopamine transporter, adding excess dopamine to the medium temporarily silences activity. So here, addition of dopamine at 100 micromolar to the medium reduced electrical activity and silenced all of the coordinated bursts, indicating that dopaminergic neurons are responsible. Washout of the excess dopamine restored the network activity. What we saw at 5 weeks was just the first glimmers of spontaneous activity. But by 7 weeks, we could see increased bursting and increased coordination. However, this activity remains very slow compared to what dopaminergic neurons can do in a monolayer culture, as you'll see in the next slide. So my colleague Michael Durr took a different approach to recording activity of these iPSC-derived dopaminergic neurons. As I told you about with the FOXA2 OTX2 staining, Michael performed floor plate specification and expansion in 3D for 11 days before dissociating the organoids and replating them onto the M8 surface. So when that specification and expansion are performed in 2D, the neurons produce rhythmic coordinated bursts by 6 weeks of differentiation, which you can see in the raster plot on the left. However, specification and expansion of floor plate in 3D produces stronger and more frequent bursts before 5 weeks of differentiation, as you see in the raster plot on the right. So we're getting more activity in a shorter amount of time. Michael also compared the mean firing rate, the synchrony, and the fraction of active electrodes for neurons replated from 2D or 3D cultures. In each of 2 cell lines, specification of floor plate in 3D led to higher mean firing rates, greater synchrony across electrodes, and activity across a wider area of the well. Also, if you look carefully at these graphs, each of these characteristics reached its maximum earlier in 3D specified dopaminergic neurons. So as you've seen, we have not yet recorded comparable activity within intact organoids. This may just be reality, or it may reflect poor contact between the organoids and the surface electrodes. Nevertheless, we have evidence that organoid culture promotes dopaminergic maturation. So I want to briefly discuss the possibilities of using midbrain organoids to model Parkinson's disease. So even among the most elderly adults, the rate of Parkinson's is just under 2%. So we cannot model the disease with just any pluripotent stem cell line. But numerous mutations have been identified in the genome which greatly increase the risk of the disease. By incorporating these mutations into our model, we have a better chance of observing disease-like phenotypes in vitro. Many mutations that are common among Parkinson's patients occur in the genes for alpha-synuclein and LRRK2. The alanine 30 to proline mutation in alpha-synuclein leads to protein misfolding, and misfolded alpha-synuclein is a major component of the Levy bodies found in degenerating dopaminergic neurons upon autopsy. LRRK2 mutations are the most common genetic cause of Parkinson's disease, and the glycine 2019 to serine mutation is found in many familial Parkinson's cases and also about 1% of sporadic cases. So each of these mutations were engineered into the same iPSC line using CRISPR-Cas9 technology by Thermo Fisher's custom services team. After introducing the single base pair changes, the cells were sorted to obtain clonal lines. Sequencing demonstrated that the desired single nucleotide polymorphisms were homozygous in anywhere from 4% to 48% of the clones. So our next step is to test whether these disease-related mutations make the cell lines more susceptible to stress. First, we can test for a phenotype of these mutations in the iPSC lines themselves. Numerous compounds will increase stress and trigger apoptosis in iPSC or progenitor cells. Two that are very commonly used are staurosporin, a broad-range protein kinase inhibitor, and hydrogen peroxide, which induces oxidative stress. We use staurosporin to test for the phenotype of the alpha-synuclein mutation in the iPSC line, as I'll show you in a moment. Second, we can look for disease-like phenotypes in mature midbrain organoids. Here, the phenotype that we want to see is selective apoptosis in dopaminergic neurons, while other neurons and non-neuronal cells are spared. A number of compounds are frequently used for inducing this phenotype in animals or in culture, including 6-hydroxydopamine, which is specifically taken up by DA neurons, Rotenone, which inhibits mitochondrial complex 1, MPP+, which is a toxic metabolite of MPTP and induces reactive oxygen species, or MG132, which inhibits the proteasome. That's just a few among many compounds that are frequently used. I'm going to show you a bit of data using rotenone as our stressor. But first, we're going to test for an effect of the A30P mutation in the iPSC themselves. So I previously described how we expand PSC in suspension culture. Here's a picture of the spheroids of undifferentiated iPSC. We can induce apoptosis in these by adding staurosporin and detect its effect with a green dye called cell-event caspase 3,7 that fluoresces after activation by caspase 3 or 7 that are regulators of the caspase activation apoptosis pathway. So exposure to a range of concentrations of staurosporin looks like this. This is the response of the cell event dye. And when we quantitate that fluorescence in live spheroids, we get curves like this. So here we can see that the A30P mutation is shifting the ED50 for staurosporin. Next, we're going to try to perform a similar assay in more mature midbrain organoids. So, these are midbrain organoids generated from alpha-synuclein A30P mutant and wild-type lines after 3 weeks of differentiation. These were exposed to rotenone for 24 hours, followed by the cell event caspase dye. Then the organoids were stained with TH antibody and imaged as whole mounts on the CX7 high content system. The magenta color is the TH staining in dopaminergic neurons, while the green is the caspase dye. You may notice that the extent of the TH staining is progressively reduced with increasing drug concentration, while the caspase dye increases, but neither of these measures declines to 0 on either end of the scale, which affects our quantitation. When we measure either caspase activation on the left or TH positive neuron degradation on the right, singly in whole organoids, we can see that it's difficult to fit the typical sigmoidal dose response curves. In this case, the wild type cell line represented by the blue is a little better behaved in fitting that sigmoidal model than the mutant. This tells us more experiments are necessary. Alternatively, we can attempt to measure the activation of caspase specifically in the dopaminergic neurons alone. To do so, we've used an antibody specific for activated caspase 3. These are whole mounts of 3-week-old organoids stained for tyrosine hydroxylase and active caspase 3 and cleared with the cytovista reagent and again imaged on the CX7. It's not particularly easy to co-localize the two antibodies. The strategy that we've used is on the next slide. So we try to focus on single cells within these densely packed organoids by segmenting the images on DAPI, as you see in the image in the center of the row, and exclude larger areas where DAPI-positive nuclei overlap. Then we score each of those segmented particles for the intensity of caspase-3 and the intensity of TH. This is how it looks if you were doing this on a Z-stack projection. But what we're actually doing is the same process on every optical section. And this allows you to more narrowly focus on the cells that are in focus in each section. It's very data and labor intensive. So given the slow progress on this, we're starting to focus on work in cryosectioned organoids instead. So here we are looking at 5-week-old organoids, the same age at which we can see the first glimmers of electrical activity. And the staining here is TH in green for the dopaminergic neurons, and red is the active caspase-3. We can see that there are TH-positive neurons below the surface of the spheres, as I described before, where in the presence of high concentrations of the rotenone on the right, you can see that there's nuclear fragmentation and a great deal of caspase activation close to the surface. But in the untreated spheroid on the left, we still get a fairly high background of active caspase-3. As you have probably read recently, most organoid cultures are under a lot of stress due to lack of oxygen and mechanical stress from being stirred. So that is probably contributing to our high background here. And one of the projects that we're working on is maintenance media for organoids that will help to reduce that stress. If we look at the numbers of co-localized TH neurons with active caspase-3, out of cryosections we can start to get decent numbers, but the high background again is making curve fitting difficult. So that's where we are to date. I just want to run through a few conclusions from this work. First is that the Thermo Fisher kit for 2D directed differentiation can be used to generate midbrain organoids. And we found that suspension culture of pluripotent stem cells is an excellent starting point for 3D cultures, partly because there's no adaptation required to get into 3D when you're already in 3D, and partly because the suspension culture allows you to scale up quite a bit. Also, we find that the floor plate specification and expansion is quite accelerated in 3D compared to 2D. Furthermore, the maturation of the dopaminergic neurons themselves is faster in 3D. Doing the floor plate specification in 3D gives you earlier and stronger neuronal activity than 2D specification. And we found that seeding of the organoids in the uncloned Sphera U-bottom plates with 2% Geltrex accelerates the TH neuron maturation as indicated by neuromelanin and electrical activity. And finally, Parkinson's disease modeling in 3D organoids is possible. With the iPSC spheroids incorporating mutations made by CRISPR-Cas9, we can show effects of mutations in iPSC. The more mature organoids are good for demonstrating functionality of TH-positive neurons, but the assays are much more difficult to do and are very low throughput. So that is all I have to say, except for thank you very much for listening to this seminar. I hope you all stay well, and I'd like to open it up to any questions.
Thank you, Dr. Josephson, for your informative presentation. We will now start the live Q&A portion of our webinar. Now to our audience, if you have a question you would like to ask, please do so now. Just click on that Ask a Question box located on the far left of your screen, and we will answer as many of your questions as we have time for. Okay, let's get started. We have some great questions coming in. Dr. Josephson, our first question. Can this system produce organoids at a large scale?
So the system as we have it produces organoids at what I'm going to call medium throughput. The throughput is certainly higher than if you are encapsulating organoids, which is very labor intensive, but it's not as high throughput as if you were going directly from the PSC suspension culture through bulk specification of spheroids into organoids. So we would like it to get to be high throughput, but we're working on that.
Very good. Now, our next question. Do you have any products for organoids of other cell types or tissues?
So there is a large effort within Thermo Fisher to produce products for a variety of organoids, from different types of brain cells, liver cells, cancer organoids, cardiac organoids, and for the most part, those projects are all in process.
Okay, Dr. Josephson, can Thermo make organoids for me?
So I mentioned that we have a custom services team in Carlsbad, California, and they have collected all of the protocols from various people working on organoids within the company and are ramping up to make organoids as a custom product. So yes, I would say contact your sales rep, ask them about custom services.
Very good. And we have time for one more question. How big do your organoids get?
Well, my organoids start out at about 200, 250 microns when I begin the specification. They grow to approximately 1 millimeter in diameter by the time I'm doing my assays. So as you saw in the MEA pictures, they just about cover all of the electrodes in a 48-well MEA.
Thank you, Dr. Josephson. Now, before we go, I'd like to thank our audience for joining us and for their interesting questions. Questions we were unable to answer live today, and those submitted during the on-demand period will be addressed by Dr. Josephson via the email address you provided at the time of registration. I would like to once again thank Dr. Richard Josephson for his time today. We'd also like to thank LabRoots and our sponsor, Thermo Fisher Scientific, for underwriting today's educational webcast. This webcast can be viewed on demand, and LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. That's all for now, and we thank you for joining us. We hope to see you again soon.